10 Minuten lang kriege ich auch meine Kalorien voll. Oh shit, das hört man jetzt.
Well, thanks everybody. You buy a CD and you always wonder what's on the back side of the CD. Well, I'm on the back side of the CD. You get an extra talk today. So again, I am Dr. Richard Allen III. I'm a computational molecular synthetic virologist, and so I'm going to talk to you about these amazing rocks that live in aquatic ecosystems that I've worked on for about 2 decades. And it's going to give you kind of a preview of our past and potentially our future. So I'm going to take you on a journey today. We're going to talk more about that. All right, so this is them, folks. These are breathtakingly large carbonate structures that are made by microbes, right? And these right here are not even to scale. They're bigger than this area here that you're seeing, right? They're as big as this room, okay? So the question people ask is, what is a microbialite? These structures that are made by microbes. So here's the long tawdry definition of microbialites. They are organic deposits excreted as a result of benthic microbial communities. All right, so they bring in light, CO2, nitrate, methane, and they create these beautiful structures that you see here. Everything from a menagerie that looks like giant cabbage heads to hard, croppings of long conal structures, and they're on every place on the planet. So they're old. They were really old. And so again, ChatGPT, my graduate student looked at that, and she's an artist, but threw this together for you. So this is what the Earth looked like. So imagine an Earth to where you can't breathe, right? Most of the skies are pink from various chemicals that you can't have. There's no oxygen in this atmosphere. And this is how most life existed, right? 3.7 billion years ago. And so if we're going out to look for life into the future in something like Enceladus or Europa or Ganymede, this is what we should be looking for. Stuff like this. And your excellent work on water and analytical chemistry can help us find these new forms of life. Actually, a friend of mine, Chris Graber, out of Johns Hopkins, actually had a nanopore sequencer off a probe and was able to detect the building blocks of life, water, of all things on Enceladus, off the plumes of Enceladus. Eventually, we want to get the sequence off the plumes of Enceladus. So this is where they are. So a little bit more rocky details, if you will. So we classify these structures by the way they look, geologically. So these are microbialites. So the classic one is microbialites, right? So we've layered it, and you can kind of see, you get the laser pointer, there we go, folks. These beautiful layered structures. And these are the ones that we see 3.5 in Turkey, Australia, or in as well, 3.7 billion years ago. Next one we see are these really cool things called dendrites, projections, like your fingers. The ones that we see most commonly are thrombolites. Thrombus meaning clot, and you can kind of see there that they're clotted together, the carbonate minerals. And then lastly, we have uncolites, which are kind of spherical spinalites. And then we have lactolites, which I don't show here, which don't really have a defined structure. But here's the cool part. So every 100 grams of microbialite, you get 12 grams of atmospheric CO2. 99% of all carbon that we've ever seen in the carbon in at one point was in the lithosphere, right? And so we can sequester this carbon into the lithosphere. We can help the climate problem. So 85% of Earth's geological history, the microbialites that are present, representing the longest continuous biological geochemical cycle on the planet. So we have models on how we think they work. And so one of the models is obviously you need the chemistry, you need alkaline water, you need dissolved CO2, you need calcium, magnesium, and you have anywhere from aerobic to anaerobic plasticity at the top of the microbial map. And then you can have basically aerobic or anaerobic throughout through stratification. And these occur in lakes, they occur in marine systems, in tidal, they occur in hot springs, you name the system. As long as this chemistry is there, we'll find them. So the big one, obviously, post. So we talked about the ancient stromatolites, and so there was no oxygen in photosynthesis until about 3 billion years ago, right? You know, everyone talks about the leader, right? The terraformers of our time. And so cyanobacteria and green photosynthesis is the reason why we're in this room today, right? So they basically pull in carbon dioxide, there's alkaline water column, the cyanos and protists as well, then liquefying that through photosynthesis of the dark cycle and trap that carbonate irreversibly. The other one that we can see through heterotrophic means, there's lots of biochemistry we can talk about, folks, but I can't because we don't have tons of time. But another one that comes up, of course, is the glitches in the middle. It's basically do sulfate reduction through anaerobic means, and you basically pull down that CO2 in the form of carbonates. So when I started my PhD, they're like, these things are really rare. Back in 09, they're like, these things they're really rare, they're not a big deal, whatever. Well, they're wrong. There's tons of them, they're everywhere. Even if we find a friend, we find, hey, there's a link with freshwater corals. They're there, right? They're in hot springs, they're in marine systems, they're in hypersaline systems, they're everywhere. So the one I'm going to tell you about really quickly is the one I worked on in my PhD, called the Lake. And so we have this Mars CSA project that allowed us to have these rovers to go and grab the microbialites. And so we grabbed some sediments, some water, we did some DNA extraction, library prep, threw that on the lab. Again, I'm a data guy, computational, so you get raw data, right? You gotta clean the data. And then you gotta use my cool new tool, which, you know, same with self-promotion type of one tool to rule them all for multi-omics. So if you're interested, let me know. And so like I talked about, what was interesting about the Lily Lake is you had these very diverse structures and how they formed as a function of geologic. So you had these initial lettuce beds in 10 meters, then the cabbage heads, you had these kind of conal structures with like asparagus croppings, and then you had these large artichokes and things that about 45 meters. We also have a surrogate lake called Kelly Lake, which I won't talk about today, but it is very different from the lake. So the question we wanted to ask was simply, what was the metabolic potential, what's the functions of the DNA, and the community structure of the microbialites. Between the microbialites, the water that surrounds them irreversibly, deep, or the sediments. And those geologists will say, it doesn't matter, right? These things are not, they're there, but they're just, you know, not doing anything else, not interested, right? Well, they're wrong. And sure enough, there is a unique structure within the microbialites. From using perhaps seed, basically say what are the organisms that are there, and then also see subsystems, the functions. So not only are the microbialites uniquely compositionally different from the ground, even though they're surrounded by water, surrounded by sediment that's drowning, the microbes there are functionally different, as well as they are taxonomically different. And there's a couple of cool microbes there we talked about. Obviously, the alpha bacteria, the beta bacteria, and then the cyanos. And then again, a lot of stuff that is shared, but most of the stuff that's in the chemistry is in the microbials. So we kind of dug deeper into the metabolism. And so we found that even though this is a pristine ecosystem, they're being exposed to antibiotics and toxic compounds. And the microbialites have a protection away from that. So you can actually see the microbialites are being toxified. These kind of mitochondrial molecules, they're great at dealing with heavy metal stress, and they're able to deal with and make butyl acetone and ethanol, which we've never seen before with this metabolic activity. So again, we're trying to figure out how they make structures. So a lot of data here, we won't go into great detail. Obviously, there's heavy metals there. You can see them there, of course, metals. But what's really interesting, we saw that urease showed up. And so urease is a heterotrophic means to actually decarbonate minerals. And so I could go out and soil right over there, grab some organisms, throw them on a calcium plate, and actually decarbonate precipitation in soil. We've never seen this in microbials. And so the chemistry is pretty clear that they actually do use urease to actually help lithify the structures deeper in the microbiome. Of course, I work on viruses, which are really cool, I think. If you're interested in viruses, lots of stories. And so what we see is that the viruses in the microbiome are not very much there. Not much at all, actually. But we see tons of things that resist viruses in the microbiome. And again, don't have time to talk about it, but there's a viral mediated transition that we see between microbialites in this way as well. basically, again, showing the viruses coming from the water column, infecting microbial communities, and then the microbialites are resisting that infection. Very complicated biochemistry here, but we're able to find that we're getting more of these new metabolisms, periodic metabolism, and then heterotrophic, which is new in structures. So let's see how we do it. Go on time? So people ask, you're a virologist, right? Why are they even involved? What's the deal with viruses? And so are they even there? It's the big question. So it's like, we're not going to get any money until you show us that they're there, right? So again, here they are, beautifully in Sharpie, Australia. And kind of zoom in to a big structure like this. Okay, maybe a little bit more to see the viruses. Maybe even more. Well, there. All right. So you can see here, this is just taking microbialite, staining it with nucleic acid beads, and we're then kind of all these dots here. These are most likely bacteria, right? And these little tiny things, those are viruses, right? And we've been measuring that for a while. So it's hard to measure viruses in aquatic ecosystems. There's more viruses in your mouth right now than there are stars in the Milky Way galaxy, right? In your own ecosystem, right? So it's hard to measure them, and it's super expensive, around $30 a sample. And I thought this was absolutely absurd, right? Insane, right? And so we developed a new technique, which now we also have a flow cytometry technique. If you're interested, let me know that you can measure all your water systems. And we're really trying to figure out how many viruses are there. And so we can take this, do some quick chemistry, use some chloroform to get rid of the knock background, and we can take that and measure viruses both in the EPS, the map, and the water. We have a high throughput method now with flow cytometry. We're going to do 60 samples in 2 hours hand free. Right? And so it's super expensive, right? $30, $40, right, for these things at the end of the day. No one should be paying that amount to measure viruses. Our method, 75 cents. You got 75 cents to measure viruses? I think you do. It's even cheaper with flow cytometry. Right? So, I love calculations. How many people like calculations? I'm kind of a nerd when it comes to calculations, right? So, we measured the volume. Well, at least someone else, brilliant, like you guys that work on water. Measured the volume of the Great Salt Lake. And then we took and measured the amount of viruses, so we assumed that every virus in Great Salt Lake was around the size of a T7, which it isn't, but we'll say it is. And then we tried to measure how many viruses. We took all the viruses and weighed them. How much weight would you have? Right? Well, we weighed them, and that's around 2.2 kilograms. And you're like, well, what does that mean, Rick? Well, that's a mass of a red brick. A standard red brick. So I took all the viruses, and I measured them, and I squished them together. They weighed 2.2 kilograms, and they'd be roughly the size of a red brick. And they would fit in a gallon container. Standard gallon container, all the viruses in Great Salt Lake would fit in a gallon container. Very cool. All right, again, same as mushroom. So if you're interested in trying to analyze Linux data, got some cool tools here for you. Two are published, Meerkat and Cerebrus, the Universal Annotator, Universal Chamber Counter, and then the Titan infrastructure, which does everything. Proteomics, metabolomics, RNA-seq, you name it, does it. Lastly, you can money. And then I'm always looking for talent. I'm always interested in new people. Absolutely blessed to be here.